Okay, well, thank you, everybody. For the second session, we're going to switch focus a little bit. We've already heard something about molecular emphasis, but I think we're going to be hearing more about that in the second session, which is focused on neurobiology of disease and I think on fundamental cellular and molecular mechanisms that are ultimately relevant to understanding disease and identifying new ways to treat it. So, and in the middle of this session, we'll have lunch out in the atrium. So we have one speaker before lunch, and this will be Dr. Kewei Wang. I apologize for my Chinese pronunciation. I hope it's good enough that you know who I'm referring to. Kewei Wang from Peking University will be talking about targeted voltage gated potassium channels for therapeutic potential in neuropsychiatric disorders. Good, thank you. And you have a countdown up there.
Sure, yeah, thanks very much. I think you pronounced very correctly. First, I'd like to thank organizers from both sides and Beijing and MIT and for putting this symposium together. And also like to thank the McGovern Institutes from both sides and for sponsoring me attending this conference. What I like to do in next 25 minutes, and like to first, and I guess give you a brief introduction about what we've been doing in the lab, and also talk about one particular project that I think we advanced. I think relative in a fast pace, and then have some interesting results. Before I start to talk about the project in the lab, I'd like to first thank to all the students and just part of the list and contributed to what I'm going to talk about in next 20 minutes. In case I have to cut my talk short, and also my collaborators at different labs, at the Chai lab at NIPS, and Zhang lab at Hebei University, and also the Yang lab, our first affiliated hospital, and also the Lion and Zhang labs at the Academy of Science in different locations. So my lab has been interesting, I guess, can be described as two lines of research or two directions. And first, it's kind of we like to understand the structure and function, neurological function of ion channels, and those can be summarized as two different types of ion channels. The first is a voltage-gated potassium channels, and the second is a ligand-gated chip channels. And based on those knowledge that we like to gain, and then hopefully can guide us for next step, which is like the goal for the next line of research is to identify the pharmacological reagents that we can use for future potential therapy of neuropsychiatric disorders. In this sense, and we like to screen channel modulators from natural resources and natural products and toxins, and also like to also identify compounds from again natural products for different ligand-gated ion channel, in this case alpha 7. I don't think I have time to talk about this, and I guess my talk today is going to focus on the last part in red, and the identification of KCNQ or Kewei 7 modulators and for therapeutic potentials. A couple of years ago, we solved a crystal structure of Kewei 4 N-terminus, and then his auxiliary subunit called K-chip1, and this channel complex. It shows that this is an interesting interaction model between alpha subunit in blue and also the auxiliary subunit in gold. You can see that the interaction really shows the two contact points or the two interfaces. For the first interface, the blue part of the N-terminus of the Kv4 N-terminus really sequestered by the auxiliary subunit, and in the same time, this molecule interacting with a neighboring alpha subunit N-terminus. So it looks like a clamp. One molecule is interacting with the two parts of two subunits of alpha subunit, so functioning as a molecular clamp. Because this channel is so important, in particular responsible for initialization of repolarization of action potentials, and it changed the shape of waveforms of action potentials. And we also solved, this is what happens when the N-terminals of Kv4 or alpha subunit binds to the K chip 1, which basically the alpha 10 subunit swings upward and generating a cavity, which is for the N-terminus of Kv4 N-terminus. And that's basically this model explains many aspects of this alpha auxiliary subunit that has a multi-functions on Kv4 modulation. We also solved another subunit of alpha auxiliary subunit called K chip 4A structure. The structure shows very different. This is a stereotype image. So you can tell that the N-terminus of this K chip 4A really occupies the cavity I showed you earlier in the K chip 1, and showing that if you compare with the aligned K chip 1 or bound K chip 1 or K chip 4A, really shows the difference. This is a bound K chip 1, and then this is the first few alpha helixes occupying the cavity I was describing earlier, and basically showing that besides if you remove the N-terminus of the K chip 4A, basically the structures of the two auxiliary subunits are identical, and this cavity, this two cavity can accommodate the N-terminus of Kv4. And again, and because of K chip 4A has so much different properties when it binds to the Kv4 channels, so it explains a lot of unique features of functional differences between K chip 4A and K chip 1. Recently, we also solved a partial N-terminus of a trip V3 structure, which is an anchoring domain repeat. Although trip V1, 2, and 4 N-terminus structures have been solved by a lab nearby here at Harvard. So we show that this N-terminus anchoring repeat of trip V3 really shows that the unique difference particularly in the finger 3 loops, and this finger 3 loops that's different from other trip V anchoring domain repeats that we know so far. We also identified, because of this channel is so important, and we also found that this channel is can be activated by intracellular protons, but not extracellular protons. It is probably the first trip channel that we know that activated by intracellular protons, which has a lot of pathophysiological indications if this channel can be activated by intracellular protons. So this is inside out patch. You can see that with the increasing concentration of proton, you can see this dose-dependent increase of the channel activity, but not the channel is not activated by extracellular protons if you record the current from outside patch recordings. And as I said, this has a lot of pathophysiological indications, and this intracellular acidification then really can cause the overactive or excessive active of this channel, then cause the cell death, but can be rescued by if you overexpress the dominant active mutant in the culture. So As I mentioned, and the part of the research, we in aspects of translational, we want to identify the pharmacological agents that can modulate those channels. Particularly, we want to use them in the future for therapeutic potentials. And to collaborate with labs as Academy of Sciences, recently we identified many polypeptide neurotoxins from centipede. You can tell that this is for instance, SSM1A that can activate TRPV1 and also, but not TRPV4 or TRPA4. And we confirmed and those toxins are very selective. For instance, if you record this by whole cell recordings, then the SSM1A can really activate the TRPV1 much faster kinetics than capsaicin that we know when we eat peppers. Then we feel the spicy is because of the activation of the TRPV1 channel. But this toxin and polypeptide does not really activate the TRPV3. Okay, TRPV3. Also, we recently identified another very interesting channel blocker for TRPV3, dafnatin, and this is the plants. So you can tell that if the channel can be activated by the opener, it is not quite selective, but it is an opener for TRPV3, can be blocked by this compound and can almost 100%. Okay, so, which is very interesting, that because so far the TRPV3 channel, even modulators and particular blockers has not been identified so far. So hopefully we can use this compound to address many questions that we're interested in, and how the channel play in the role play in neurological functions and also the other related tissues. So the primary part, the part that I like to share with you is one of the projects that has been happening in the lab for last 5 years, I would say. And the guiding principle basically that guides us for this research is that, okay, this is the normal action potential, next potential that we're all familiar with. So if the idea is that, so everybody, I guess, in the audience will agree with me that hypo or hyper neuronal excitability basically defines the fundamental mechanism of neuropsychiatric disorders. So if somehow we can, for instance, this is you like to think pain, epilepsy, or schizophrenia. If we somehow can turn this situation back to this by activating voltage-gated plasma channels, then we probably can achieve some potential for new therapy. So with this in mind, we selected one of the target, and that is highly expressed in the brain, but not in other tissues. And this is a northern blot. And so those KCNQ 2 and 3 are highly expressed in the brain. And this is this is another evidence that this channel controls the memory excitability. This is action potential fired, but without reaching the subthreshold, the neuron does not or cannot fire a more action potential because of the activation of voltage-gated KCNQ or Kewei or M, meaning the same thing. So because the activation of this potassium channel, so even you stimulate it, so neuron cannot fire more action potential. But if you block it and the red arrow reduce, so and then if you stimulate the neuron, you can see the repetitive firing. And this is very much we can think this is a situation of epilepsy and pain, also the schizophrenia, if you like. And this KCNQ 203 channels basically 6 transmembrane domains and a lot of mutations, and then those squares and circles are identified from the human neonatal epilepsy. So a lot of those mutants and then can carry out into adults when about 30 or 20% of patients can have epilepsy in their later life. This is the current, so just a summary of this current, and then described as a non-inactivating voltage gate plasma channel, and activated by depolarization, and inhibited by muscarine stimulation. That's why it has the M called M current. It's a sustained current, particularly in the range of action potentials. So it has a very profound effect on neuronal excitability. It's a slow activation, slow kinetics activation, and deactivation basically function as a brake for suppression of repetitive neurofiring that I showed you earlier. But if you inhibit this current, also can lead to the excessive neuronal excitability. So to start to screen useful modulators, we first generated stable cell lines expressing KCNQ2 alone or KCNQ3, and to test that we have both target expressed in the cells, and we use the common potassium channel blockers to block the channel, because this is for both heteromeric expression of 2 and 3 is less sensitive to TEAs and than Q2 alone. So we know we got the stable cell lines expressing both of this the target. And then we set up atomic absorption rubidium effluxase in 96 format, which allows us to screen a focused library. The principle is very simple. The rubidium chloride samples are sucked into nebulizer and then into the flame and burned to generate the rubidium atoms, and can be detected at a specific wavelength at 780 nanometers. So with this, this assay is very simple. I don't want to describe more. So with more synthesis, targeted synthesis and substitutions, we came up with a series of pyrazole and pyrimidine series, and basically tested the dose response of this compound, in particular KO58, dose-dependent activation of this current, and compare with a benchmark compound called reticabine that was marketed in 2011 by FDA for partial epilepsy and treatment. So and then we because our first high throughput screening was rubidium flux, then we need to test those compounds in patch clamp. That's a gold standard of this assay. So it shows that KO58 can dramatically increase the current. Okay, dose dependently increase the current, and so this can shift the voltage dependence activation to the left. So meaning this, and then also slows down the deactivation. You can see that this fast deactivation and slow deactivation, meaning that the channel spend more time in the open state. And we also tested the selectivity for this series compound, and you can see that in particular this compound can enhance more current for neuronal type of Kewei 7.2 and KCNQ2. So since we have these openers, and then we also want to test if they can activate the KCNQ or native KCNQ M channels in DRG neurons, because DRG neurons are very important for transmitting the pain signals. So we can see that, yes, indeed, with a KO58, you can see that there's a current dramatically increased in DRG neurons. And also this current can be blocked by a specific blocker called XE991. This compound was developed by DuPont back in 1980s. The purpose of having this compound was to enhance memory, but it happened to be now to know that this is a blocker of the KCNQ channels. And so to test the in vivo pharmacology, we obviously need to know the pharmacokinetics and how long the half-life of this compound is when you inject into the bloodstream. And we got about 6 hours for half-life, and which the long enough, and then can allow us to perform the in vivo pharmacology. Yeah, this is a PK profile. I'm not gonna, in different, also the absorption in different tissues, and then the half-life, I think it reaches the peak around, I think, 7 hours. So we tested this compound first in epilepsy models, and you can tell that this is control EEG. And this is a epilepsy EEG induced by the PDZ. There's a epilepsy inducer. I'm sorry. And this is 1 time and 7 times dose. So for 6 days, six times once a day. So you can see that the hyperactive neural activity was suppressed. And then this is just summary for this compound that can really reduce the death rate compared to radicabine and or the control. And also tested different pain models. In here, in particular, the inflammatory pain models. There's a bradykinin-induced inflammation, so you can see that really compared with the benchmark compound and our Q58 theory can reduce the linking times and also the increase the pain threshold, suggesting that it has some effect on can reverse the pain sensitivity. So this is a brief summary, and I briefly introduced what we've been doing back in Beijing for the last several years, and particularly focusing on the structure and neuronal or pharmacological function of 2 types of channels, Kewei voltage-gated potassium and trip channels. And today, I just focus on one of the type of voltage-gated potassium channels in Kewei7, KCNQ. And the reason is that this channel is really brain specific, and a lot of studies confirm that it can be a good target for many neurological disorders. So we started by generating stable cell lines and screen a focus library, identify Q58 series as a novel lead compound with 2 different assays and 96 wire format and patch climate recordings, and showed some efficacy in vivo pharmacology. And then we think this the compounds we identified could be to further validate this target is in good use of new potential therapy for neurological disorders. And so with that, I'd like to thank for your attention. Thank you.
Good. Well, thank you very much. The talk is open for questions. So I have a first question. What other indications other than epilepsy do you foresee for these potassium channels?
Yes, as I mentioned, epilepsy, pain, and schizophrenia, and also attention deficit disorders, and this kind of indication for particular for openers. For blockers, I don't have time to show this, and I think obviously if you moderately increase the neuronal hyperexcitability, probably good for memory, cognition, and probably for Alzheimer's disease, but I don't have time to talk about the second part of this project, which we are also looking for selective blockers for this channel, and so, and then I guess there's a couple of papers describing the overexpression, the induced overexpression of BDNF when you have memories, have a short or long-term training, or you block this channel by blockers. But I don't have time to talk about this. Yeah.
Question over there.
Oh, maybe I just got some technical question.
Okay. Yep.
I saw one of the slides you show in terms of the concentration is around 10 micromolar. That can be a drug property, right? I mean,
No, this is
the concentration is that high. So that's one, and the other is there evidence that this compound actually is a brain penetrable?
Yeah, with a compound distribution that we tested and collected different parts of tissues and shows that it can pass the BBB. And for the concentration we're talking about, we compare with the drug that approved by FDA in 2011. It's a slightly lower than retigabin, so which we don't think that could be that's a problem. I think it's comparable for the concentration that you were mentioning.
I'm not talking about in vivo pharmacology, which I show. I mean, that could be a direct or indirect factor. I mean, in terms of cell patch or the channel, you use a reticabin.
That's correct. Yeah, reticabin.
As a comparison, I saw the concentration was pretty high. It was 10.
Around micromolar, I think 2 or 3 micromolars is comparable to around 3 or 5 micromolars for reticabin. It's not that far.
Good. Any other questions? Yeah, we have one over there.
Just a very quick question. You mentioned the Kv7.2 is a neuronal-specific subtype of potassium channel. My question is this neuronal type Kv7.2 is ubiquitously expressed in the brain regions, or do you see any brain-specific region enriched with this subtype?
In one of the slides, I showed that the northern blot, I think, from a group in Germany, they have it's not in some nucleus. I think they do have a relatively higher expression than others. So, for instance, the amygdala, I really like to think that probably play a role in mood-related behavior. So it's not ubiquitously expressed in every nucleus. It does show some different expression in different nucleus.
Okay, if there are no more questions, we'll thank Dr. Wang again.
Thank you.